Welcome back to the deep dive. Today we're tackling what many neuroscientists call the greatest bottleneck in the field. How do you translate these amazing causal discoveries you make in a mouse brain into meaningful clinical knowledge for a human being?
It's a fundamental challenge, and it's really rooted in evolution. On one hand, you have these rodent models, and they are absolutely crucial for understanding mechanism. We can go in, we can precisely manipulate a circuit, turn it on, turn it off, and really figure out the why. But at the end of the day, they are, rodent brains.
Right. And on the other side, you have all these huge human studies, clinical trials, brain scans, and they give us all the real-world variability and clinical relevance we need, but they're purely correlational. We can't just reach into a human brain and flip a switch. So the old methods of just saying, okay, mouse region X is the same as human region Y, they just fall apart.
They fall apart completely. The complexity, especially in our cortical regions, the parts responsible for all our higher cognitive functions, has just foiled any simple cross-species comparison. And that translational roadblock is why so many promising preclinical drug targets, they just crash and burn in human trials.
But the sources we're looking at today introduce something new, something ambitious, a computational framework designed to finally bridge that chasm. It's called TransBrain.
That's right. And TransBrain sounds like a sci-fi concept, but its mission is actually very concrete.
So what is its mission exactly?
Its goal is to create a quantitative bidirectional translation map, something that allows you to map whole brain phenotypes between humans and mice, and then back again. And it does this in 3 big stages. First, it builds this super accurate molecular map based on gene expression. Second, it integrates the brain's wiring diagram, the structure, to make that map even better. And third, it actually applies this new unified framework to solve real-world problems, things like interpreting mouse circuits or even understanding individual patient differences in disease.
Okay, so let's dive into that first stage then, the molecular foundation. If the human cortex is so complex, how did they get past that first big failure point? Where did the traditional methods go wrong?
The primary failure was really about the data. It was just inadequate. Previous attempts just didn't have human transcriptional data that was both high resolution and high volume.
So it's like trying to draw a detailed map of a huge, complex city, but you only have a few blurry satellite photos.
Exactly. So TransBrain's first innovation was a data integration strategy. They merged 2 massive but complementary human datasets. First, you have the Allen Human Brain Atlas, or AHBA. That gave them incredible spatial resolution, great detail, but not a ton of samples.
Okay, so the detail is there.
Right. And they combined that with a huge single-nucleus RNA sequencing dataset. Now, this gave them millions of individual cellular profiles, so massive volume, but not great spatial information at first. By merging them, they created this incredibly rich training pool that had both the high resolution and the statistical power they needed.
That makes sense. It addresses the scale. But even with great data, the cortex and the subcortex are so different, right? If you feed them into the same algorithm, wouldn't the model just get confused?
And that is precisely what happened in earlier attempts. It's like using a single general doctor for every possible medical issue, from heart surgery to a skin rash. It kind of works, but it's not specialized.
So what was TransBrain's fix?
They introduced what they call the detached deep neural network, or DNN model. So instead of one single shared model, they trained independent modules. One specialist module just for the cortex, and a totally separate one for the subcortical regions.
Ah, I see. So you're giving the complex cortex its own specialized analytical tool because its organizational rules are just so different from, say, the brainstem. What was the actual payoff for doing that?
Oh, the payoff was staggering. This detached approach massively improved the model's ability to correctly find the homologous or corresponding brain regions in the cortex. It improved mapping accuracy by a stunning 89.5% over using the original data.
89. That's a massive jump. That really suggests that researchers were flying blind before, just using rough estimates.
It fundamentally changed the game for cross-species cortical comparisons.
So with these new, hyper-accurate maps, what did they actually learn about the fundamental similarities and differences between our brains and a mouse's?
It showed us where evolution drew the line, so to speak. The analysis found 2 huge, evolutionarily conserved transcriptional gradients. They call them PC1 and PC2. And these 2 gradients accounted for over half of all the gene expression variants. They're like the fundamental shared blueprint.
So the basic blueprint is still mostly the same.
Mostly, yes. But then they found a third component, PC3, that was clearly species-specific. This gradient lined up perfectly with the brain areas we know expanded in primates, the very structures that make our brains so complex.
But that's the primate-specific upgrade, in a sense.
You could call it that. It suggests that while the fundamentals are conserved, this third gradient represents a whole transcriptional program that's unique to us primates.
Okay, so before we move on to the structure, let's just quickly recap what we've learned so far. First, TransBrain solved that cortical complexity problem by combining two powerful datasets, the AHPA and the snRNA-seq data.
Right, for both detail and scale.
Second, the key innovation was that detached model, which trained the cortex and subcortex separately and gave them that massive 89% accuracy boost.
A huge improvement.
And third, this new map confirms there are two major conserved organizational patterns, but it also highlights a third primate-specific one that helps explain our evolutionary divergence. That molecular foundation sounds really solid. But a brain isn't just a bag of molecules, right? It's a network. It has structure. Why wasn't transcription alone enough?
Because brain function is constrained by anatomy. How information flows is dictated by anatomical connectivity and hierarchical organization. Think about it like a city's road network. Knowing what kinds of businesses are in two different neighborhoods isn't enough to understand the city. You also have to know if there's a highway connecting them.
And those highways are conserved across species.
Highly conserved. They govern the flow of information.
So they needed to merge the molecular map with this structural map. How do they actually do that?
They shifted to a graph-based approach. Imagine they built this one colossal universal map, a heterogeneous graph, where every single point or node is a region in either the human or the mouse brain.
Okay, so all the regions from both species are in one big network. How did they define the connections?
In two ways. The first were the intraspecies edges, so the connections within each species. For the human side, they used diffusion tractography, which estimates the brain's major white matter pathways. For the mouse, they used the gold standard highly precise viral tracer data.
So that's the internal wiring diagram for each. But how did they build the bridge between the mouse nodes and the human nodes?
That's where the molecular accuracy from the first stage comes in. The cross-species edges were weighted by those really refined transcriptional similarities, but, and this is the crucial biological safeguard, they were strictly constrained by coarse anatomical hierarchies.
What does that mean exactly?
It means they pruned connections that were biologically impossible. You wouldn't, for example, allow the model to connect the mouse olfactory bulb to the human visual cortex, even if they happen to share a few genes by chance. It keeps the model grounded in reality.
So using known biology to clean up the computational noise, that's clever. Once they had this massive, highly constrained graph, how did they pull out the unified map?
They used a machine learning technique called graph representation learning with an algorithm called node-to-vec. You can think of it as teaching the algorithm a shared abstract language. This process embeds every single brain region, human or mouse, into a unified latent vector space. Now, every point in that abstract space represents a region's total identity, its genes, its neighbors, its place in the hierarchy, everything.
And that shared language, that's what makes the translation possible.
Yeah.
So did adding all that structural information actually help?
It was absolutely critical. When they checked, the correlation with known human structural connectivity just jumped. Using just the transcriptional data, the correlation was pretty weak, around 0.32.
Yeah.
Once they added the structural and hierarchical constraints from the graph, that correlation shot up to a much more solid R equals 0.59.
Wow, that's a huge increase. It really shows that the brain's wiring provides essential information that you just can't get from genes alone.
It's an essential constraint. And all of this leads to the final mechanism, which is dual regression. Because both species now live in this shared space, TransBrain can perform bidirectional mapping, projecting findings seamlessly from one species to the other.
All right, now we get to the applications. Let's see this translator in action. Case study 1 was about resting state networks, right? The brain's fundamental communication pattern.
Yes, and they quantitatively assessed how well these functional features mapped across species. They found some expected things, like human gradient 2 corresponding really strongly with mouse gradient 2, but the most insightful result was a complete surprise. Something that forces us to rethink a little bit about mouse evolution.
Oh, and what was that?
TransBrain found a really strong and significant correspondence equals 0.57 between the complex human ventral attention network, or VAT, and the mouse salience network.
And the VAT network in humans is for detecting new things and shifting our attention, right? Functions we think of as being highly evolved.
Exactly. And that's why it was such a surprise.
Why?
Because the prevailing wisdom was that mice lacked any clear evolutionary precursors for the specialized VAT network we see in primates. This quantitative match suggests that the fundamental principles of attention and salience processing are far more conserved and maybe more complex in the mouse brain than we ever appreciated. It reframes how we can use mice to study attention disorders.
That's a huge biological insight to come from a computational model. Okay, let's move to case study 2. Taking causality from the mouse lab and translating it into complex human behaviors. This is the holy grail.
It really is.
Wow.
Here, they leveraged mouse optogenetic fMRI studies. Researchers can genetically modify specific mouse neurons to respond to light. It lets them literally flip a circuit switch on and off.
So they can stimulate, say, the insula in a mouse, and then measure the whole brain fMRI pattern that results from that specific causal event.
Precisely. They have a clear causal network pattern, and that's where TransBrain steps in.
It takes that mouse pattern and maps it to the human brain.
It maps it onto the virtual human brain space. Then they compared that predicted human activation map against the massive neurosynth database. And neurosynth is basically a giant map that links thousands of human fMRI studies to specific cognitive functions and behaviors.
So what did the mouse circuit for the insula predict in humans?
When they mapped the pattern from stimulating the mouse insula, it accurately correlated with human functions like decision-making, risk assessment, addiction, anticipation, and pain processing.
That is a near-perfect match for the established human role of the insula. Unbelievable. What about the other example, the dorsal rafin nucleus?
The DRN, yeah. It's a major hub for serotonin. When they mapped its stimulation pattern, the predicted human network correlated strongly with functions like cognitive monitoring, valence processing, and mood regulation.
So this is the first time a causal circuit manipulation in a mouse has been quantitatively linked through a model to these complex human mood and cognitive functions.
The first time. It's a massive proof of concept.
Let's just pause to recap that. First, integrating the structure was absolutely key, boosting the map's accuracy up to that R of 0.59.
Yes, essential.
Second, the functional analysis revealed complex conservation, including that really surprising game-changing match between the human VAT network and the mouse salience network.
Right. It challenges old assumptions.
And third, TransBrain really proved its worth by translating causal mouse circuit activity like stimulating the insula or DRN into accurate predictions of complex human behaviors like addiction and mood regulation.
Yep.
Okay, now for the final and maybe most clinically urgent application, linking specific genetic mouse models to individual patient outcomes in a disorder as complex as autism spectrum disorder. How does TransBrain enable precision medicine here?
Well, ASD is defined by its incredible variability. One patient's brain can look wildly different from another's, even with the same diagnosis. TransBrain tackles this by comparing specific genetic mouse models, which represent specific biological causes, directly to individual human patients.
So how do they quantify those individual differences on the human side?
They used a technique called normative modeling on brain imaging data from the ABIDE-I cohort. In simple terms, this establishes what a typical brain volume should be for any given age and sex. Then they can calculate a D-score, which is just a measure of how much a specific brain region in an individual ASD patient deviates from that norm.
So every patient gets a unique deviation fingerprint of their brain structure.
That's a perfect way to put it. Then they created the corresponding fingerprint for the mouse models.
Wow.
They looked at structural changes in several genetic mouse models relevant to ASD, like DVL1, MEGL2, and SHANK3. They quantified the volume difference between the mutant mice and control mice using Cohen's D-scores, which is just a standardized way to measure the size of that effect.
And TransBrain's job is to be the ultimate pattern matcher. It takes the structural pattern from a specific mouse, maps it into human brain space, and then calculates a risk score for every human patient based on how well their personal D-score pattern matches the mouse pattern.
Exactly. And they did this crucial validation where they also calculated a second risk score based purely on where the human genes themselves are expressed. The fact that these two scores correlated so well confirmed that TransBrain was translating the underlying biology correctly.
And which mouse model turned out to be the most compelling match?
The MEGL2 mouse model. It showed the highest correlation between the structural pattern in the mouse and the gene expression pattern in the human brain at R equals 0.45.
That's a tight link. It means the brain changes from that one gene mutation in the mouse are spatially aligned with how the gene is normally expressed in humans.
A very tight link.
And so, the final piece of the puzzle. What was the clinical outcome for those human patients whose brain patterns so strongly matched that MEGL2 mouse model?
This is the most crucial translational finding of the whole study. Those individuals whose brain deviation patterns most closely mirrored the Magel-2 mouse. They exhibited significantly poorer fluid intelligence quotient, or FIQ, scores.
Fluid intelligence.
Yeah.
Our ability to solve new problems and use logic.
Exactly. This demonstrates a conserved gene-brain behavior relationship that spans from a mouse model all the way to a clinical outcome in a human patient.
And which parts of the human brain were implicated in that specific pattern?
The alterations from the Magel-2 mouse mapped to the human anterior cingulate cortex, the medial prefrontal cortex, the orbitofrontal cortex, and the insula.
All regions that are heavily involved in cognitive control and decision-making.
Which aligns perfectly with a deficit in fluid intelligence.
So to pull this all together, TransBrain is this comprehensive, objective, and bidirectional tool. It provides a shared language built on molecules and structure that finally moves neuroscience beyond just qualitative comparisons and into quantitative translational precision.
It's transformative, but it's also really important for you, the listener, to remember the limitations. The authors are clear that TransBrain is best for system-level translation understanding overall networks. Trying to infer homology for one tiny, isolated region still requires a lot of caution.
And like any model, it's only as good as the data you feed it. Human diffusion tractography has known biases that can influence the map. But these are solvable problems.
Absolutely. The framework is built to incorporate new data, and the obvious next step is integrating other things, like proteomics and epigenomics, to make it even more refined.
So what does this all mean for the future of clinical research? I mean, given that the structural patterns from the Magel-2 mouse actually predicted lower fluid intelligence in a specific subgroup of patients with autism, how could this revolutionize clinical trials? Could we use this to identify the specific patient biotypes who are most likely to respond to a new therapy developed in an animal model? If we can isolate the genetic circuit for a specific deficit in a mouse, how quickly can we translate a targeted treatment to the exact group of human patients who need it most? That's really the question TransBrain is challenging us all to answer.
It forces us to think about disease not just by its symptoms, but by its underlying circuit mechanisms.
A compelling thought to end on. We'll see you soon for the next deep dive.